Okay, so here's our kidney. Here's the capsule. And this is the cortex all the way around. Here's the medulla. So here are the pyramids of the medulla. Notice we got this nice pyramid shape. Okay, and the nephron is located part in the cortex and then part in the medulla. And here's one of the nephrons here. So down the collecting duct into the medullary collecting duct, all these nephrons, 1 million of them, they all drain into this renal pelvis. And they all drain first into something called the minor calyx. So right below the pyramid, if I was to touch right here, that would be a minor calyx. Where you have 2 minor calyxes drain, we call that a major calyx. Okay, so minor calyx, major calyx. And then here's the renal pelvis, which is the whole thing that it drains into. Okay, and then here's the ureter coming out. Here's the renal artery, renal vein. There's probably something else I need to say here, but I'm going to zoom in on this nephron now. Okay, so here's the nephron, and if we zoomed in, and it's kind of nice they have the color coding. So here is the cortex, and then here's the medulla, and here's the afferent arteriole going into the glomerulus, and then the Bowman's capsule is around, and the whole thing is called the renal corpuscle, and then you have the efferent arteriole leaving. So this is the location where filtration happens. If you want to zoom in on that, this is what it would look like, okay? And I know this is the afferent arteriole because it's color-coded, but even if it wasn't color-coded, I would still know this is the afferent arteriole because it has those specialized cells called the juxtaglomerular cells, okay? So it kind of like sticking out a little bit, and you also see the macula densa cells. So I know this is part of the distal convoluted tubule, okay? So distal convoluted tubule with the macula densa cells, and then the juxtaglomerular cells of the afferent arteriole, okay? So the afferent arteriole comes into the glomerulus, about 1/5 of the blood comes in here and gets filtered, okay? So what gets filtered out? The things that get filtered out are the small things, okay? Those are the ones that go down the drain, okay? All the rest of the things bigger than 50 kilodaltons are gonna stay in this and go out the efferent arteriole, okay? So what is that? That's like red blood cells, white blood cells, big proteins, all sorts of things like that. What goes down the drain? What goes into this the renal capsule? Water, glucose, ions, toxins, urea, all sorts of things that are small, okay? So small things go in here, and this is the proximal convoluted tubule. So if we were to go back here, this light green is the proximal convoluted tubule. So you kind of just follow it around, and that's where the proximal convoluted tubule is. So what happens in the proximal convoluted tubule? Bulk reabsorption. What we mean by that is everything except for the toxins get reabsorbed. Water, glucose, ions, any small amino acids and small proteins. That's another test that they do. They want to make sure there's no protein in your urine to make sure your kidneys are functioning correctly. So this is where they must be bulk reabsorbed, okay? At this point, when the filtrate, which is the stuff going down the drain, once the filtrate enters the descending limb of the loop of Henle, it is isoosmotic, okay? It's isoosmotic because there's really no change in the concentration of water or ions. It's approximately equal, okay? So isoosmotic going down here. As we go down, there's an increase in the osmolarity of the medulla itself, okay? So going down, we have an increase in the osmolarity of the medulla. We also have an increase in the filtrate osmolarity as well. So it becomes hyperosmotic. Okay, why does this happen? Well, because we have water reabsorption. The descending limb is permeable to water. Okay, so through passive transport, the water exits the descending limb into the tissue of the medulla. Okay, so as we're going down, more and more water is being reabsorbed until you get to the bottom of your loop of Henle, and that's where the filtrate is very hyperosmotic. Okay, so now as we go up the loop of Henle, we're going to have active transport of salt. Okay, so active transport of sodium chloride. So it's getting pumped out of the filtrate, out of the tubule, into the tissue of the medulla. This is what creates that hyperosmotic gradient. Okay, so the more sodium that is being pumped out, the higher the osmolarity of the tissue in the medulla. Okay, and at the end we get actually a hypoosmotic filtrate because so much salt has been pumped out. Okay, so this is a way for juxtamedullary nephrons, which is the one that I'm showing right here, to really concentrate the urine. Okay, we can concentrate the urine, we can take all that water out, as much of the water out as we need to keep in the body. Okay, so as it's coming out, it goes to the distal convoluted tubule. Distal because it's last, and convoluted because it's convoluted. This part is regulated by hormones, so ADH, vasopressin, and aldosterone. And these are going to increase water retention and therefore increase blood pressure. We also have secretion in the distal convoluted tubule. Secretion of hydrogen ions and potassium, and reabsorption of sodium. And so after that happens, we go to the cortical collecting duct. Cortical because it's in the cortex and it's collecting. You notice that you have a bunch of these nephrons that are draining into the same cortical collecting duct and medullary collecting duct. And so they all drain into this collecting duct. Here we have, in the cortical, we do have regulation with ADH and aldosterone. And in the medullary collecting duct, we mostly have reabsorption of urea, some water. Okay? So all of these drain down, and they drain down into your minor calyx, your major calyx, and then your renal pelvis. So one more thing, okay? So one thing I didn't talk about were the capillaries, okay? So the capillaries are shown here. You have the vasorectal capillaries associated with the loop of Henle, and then you have the peritubular capillaries that are associated with the proximal and distal convoluted tubule. So these are the capillaries that are going to get reabsorbed into, okay? The other thing I wanted to mention is the different types of nephrons. So you have a juxtamedullary nephron, and then you have a cortical nephron. The cortical nephron is located mostly in the cortex, whereas the juxtamedullary nephron goes all the way into the medulla. So that one is a juxtamedullary nephron, and that one's not a very good cortical nephron. It should kind of maybe come a little less. I want to talk about the cells too, the cells that are located in different parts of the nephron. So in the proximal convoluted tubule, you're going to have lots of microvilli. Okay, so it's going to look kind of fuzzy on the inside. Notice the lumen is a little bit smaller. In the ascending, the last part of the ascending limb, and then the distal convoluted tubule, you'll have, again, both of these are simple cuboidal, but no microvilli. Why do you need microvilli? To increase surface area, to increase reabsorption, bulk reabsorption in the proximal convoluted tubule. You also have, in this thin part, simple squamous. Okay, so the part of the loop of Henle has simple squamous. Here's another model of the Bowman's capsule and the glomerulus. Notice they didn't label which one's which. How do I know which one's which? We look at the macula densa cells that are associated with the distal convoluted tubule. They are going to be in direct contact with the afferent arteriole. Okay, so direct contact with the afferent arteriole, which has the juxtaglomerular cells, which secrete renin, which regulate blood pressure. That's it.